Hi dear students, so today let's talk about phytopathogens. So what are phytopathogens or what is phytopathology? Plant pathology is also termed as phytopathology and it is the scientific study of diseases in plants caused by pathogens which are infectious organisms and environmental conditions. So phytopathogens are the organisms that will be resulting in plant diseases. Organisms that cause infectious diseases include fungi, oomycetes, which are fungi-like organisms, bacteria, viruses, viroids, virus-like organisms, protozoa, nematodes, which are small worm-like animals, and phytoplasmas are obligate bacterial parasites of plant phloem tissue and other parasitic plants. Plant disease is an impairment of normal state of a plant that interrupts or modifies its vital functions. It also leads to the reduction in yield and it also affects its appearance. Many plant diseases are caused by pathogens and a plant may be said to be diseased when there is a harmful deviation from normal functioning of the physiological processes. The classification of plant diseases based on causal agents have been shown here. In that, the first one is non-infectious plant diseases. They are not associated with any animate or viral pathogen. So they cannot be transmitted from an infected plant to a healthy one. They are due to disturbances in the plant body caused by lack of certain inherent qualities, by improper environmental conditions of soil and air, and by mechanical influences. And some of the examples are the low or high temperature, unfavorable oxygen levels, water levels, hail, wind, air pollution. You can see the effect of these environmental conditions on the plant, like the lightning injuries, due to wind damage, due to the presence of air pollution toxicities. The next one is the infectious plant diseases. These are the diseases caused by pathogenic organisms such as fungi, bacteria, virus, nematodes, and even some plants can be pathogenic. They obtain nutrients, water, and everything they need to reproduce from their host. Fungus and viral pathogens cause many plant diseases, whereas bacteria and nematodes pathogens are few in it. Some pathogens can infect several kinds of plants and others are specific to a particular type of host. Pathogens such as fungi and bacteria differ in their ability to survive, spread and reproduce. For a pathogenic disease to occur, three conditions must be met. The first one is that the host plant must be susceptible. Second one is an active living pathogen must be present. And the third one is that the environment must be suitable or favorable for the disease development, and all these factors must occur simultaneously. If one of the factors is absent, means the disease will not be occurring. Since the disease is a result of simultaneous interaction between environment, host and pathogen, the more the circles overlap, the more severe will be the disease. Disease cycle. The sequence of events from a pathogen's survival to plant disease development and back to pathogen cycle is called the disease cycle or the pathogen's life history. So why we need to understand about the disease cycle? By knowing the chains of events that contribute to a disease, we can find the weakest link and take measures to break the cycle so that we can prevent the plant from diseases. Most pathogens must survive a period of adverse conditions, usually winter, where they do not actively cause disease. Let's see the symptom of fungal diseases. First one is mildews. Means white, grayish or brownish purplish patches will be appearing on the leaves, stems or fruits, as you can see. Next one is rust. Relatively small pustules or spores usually breaking through the. And you can see rusts. These are usually seen in the back of the leaves. They are pustules of spores and it break through the host epidermis. Then there are smuts. They are purplish black or black dusty masses. And there are white blisters which break open and it expose a powdery mass of spores. Then you can see scabs. It is usually found in potatoes. It has having a crust-like lesion and a freckled appearance. Then there is sclerotia. It's often a hard mass of dormant fungal mycelium and it is often seen in black or dark brown or purplish in color. Then you can see blotches. It is also a superficial growth, giving the fruits a blotched appearance. Seen in apple fruit. Then there are fruiting bodies, also which is developed by fungi, rotting fungi, and it's mainly seen in wood. Then there are tar spots, black coated fungal bodies, which appears like tar. So we saw the symptoms of fungal diseases. So now let's check into symptoms of bacterial diseases. The first one is exudations. In this, masses of bacteria ooze out from the affected organ and appear on the surface as drops or smears. As you can see here, these are bacterial leaf blight on wheat. Here the bacteria ooze out and it comes in the form of appear in the form of a drop. In this, bacterial slime stream out of the xylem tissues. Besides the symptoms that we discussed earlier, there are other symptoms which appears when a plant is affected by a disease. And this disease causes marked changes in the form, size, color, texture, attitude, habit of the plant or some of its organ. Two or more of these changes may occur in the same host organ as effect of the same disease. And these changes result from the presence and activity of some pathogenic organism and also from the reaction of the host tissue against them. The pathogen can be found within the affected tissue or upon the surface. Like that, there is color changes, discoloration of the plant, that is sometimes it is termed as chlorosis. When green colored plant is changing to yellow, it's termed as chlorosis, and when green colored plant become colorless, it's known as albinism. And chromosis refers to the change of color from green to red, purple or orange. And there can be overgrowth. Abnormal increase in the size of one or more organs of the plant is also seen when the plant is affected by diseases. This is usually the result of stimulation of the host tissues for excessive growth. It may be due to hyperplasia or hypertrophy. Hyperplasia is abnormal increase in size of the plant due to increase in the number of cells. That is, when the number of cells is increased, there will be a significant increase in size of the plant organ. And in hypertrophy, increased size of the organ is due to increased size of the cells. It's not the number, but because of the increased size of the cells. It is termed as hypertrophy. The next one is atrophy or hypoplasia. Here, inhibition of growth occurs resulting in stunting or dwarfing. And atrophy is degeneration of cells and organs, whereas hypoplasia is the abnormal decrease in the size of cells and organs. The next one is necrosis. It is the death of the tissues and organs due to parasitic activity. Then wilt. Wilts mean it is a drying or wilting. Drying and wilting means the same thing. Here the leaves and succulent parts lose their turgidity and become flaccid and they droop. And dieback, it is a dying of plant organs, especially stem or branches, backwards from the tip. Now let's see the control measures for plant disease, and it is being divided into exclusion, eradication, protection and immunization. So the first one is exclusion. This method includes the quarantines, inspections and certification, and to prevent movement of diseased plant material into a particular country, state or geographical area where the disease doesn't exist. Quarantines are regulations for forbidding sale or shipment of plants or plant parts. Next one is avoidance. If a disease doesn't occur in an area, you may be able to avoid its development on your plants. Planting certified virus-free stock is a good way to avoid viral diseases, and root rots can be avoided by avoid planting in poorly drained soils. Delaying planting until soils are dry and warm can avoid damping off of vegetable seeds. And avoid wounding plants when pruning. Pruning means a selective removal of a certain part of the plants, or when using any equipment, we have to assure that no wounds are made to the plants. And using good horticultural practices such as proper fertility, pruning, watering to ward off infections. In eradication, the first method is crop rotation. It is common in both commercial and home gardens involving planting of different crops in a given location each year. It can reduce soil populations of fungi and nematodes. And the rotation will only work if you plant the known host. And to practice rotation, you need to know the pathogen and which plants are exposed. The next method in eradication is sanitation. Removing plant debris is a very important practice to avoid pathogen invasion, and you have to dispose of this debris by burning, burying or hot composting. And the next method is elimination of alternate host. Certain pathogens complete their life cycle on two or more hosts. For example, the organism that caused rust, only one of which may be a crop plant. And eliminating the alternate host may reduce the pressure from these diseases, since such pathogens cannot complete their life cycle without the alternate host. So when the pathogen cannot complete their life cycle, it cannot initiate the disease in plant. These are some of that methods earlier mentioned in eradication, that is the removal of infected plants that is nearby, removal of alternate host, and pruning of host to remove infected areas of the plant. And the infections and infestations can be also prevented by using chemicals or fungicides. Certain fungi and nematodes can be reduced by fumigating or steam sterilizing the soil. Fumigating also involves application of chemicals. Heat treatment is usually used to eliminate viruses from propagation material. This picture shows the steam treatment of soil. And solarization is a non-chemical environment-friendly method for controlling pests using solar power to increase the soil temperature to levels at which soil-borne plant pathogens will be killed or greatly weakened. And the third method in control measure is resistance. That is to generate disease-resistant plant varieties through genetic mechanisms or recombinant DNA technology. Resistance, immunity, tolerance and susceptibility are the terms that describe a plant's genetic makeup and its reaction to plant pathogen. Planting resistant cultivar is the easiest means of disease control. If a plant never becomes diseased, the term immune is used, and the word tolerant describes a plant which may become diseased yet grow much like a healthy plant. And protection means treating a healthy plant before it becomes diseased. Both chemical and biological means are available. Chemical protection is a common technique which involves fungicides. And the biological means we can use another bacterium which will give protection from the pathogenic bacterium.